I am Zhao Fu from University of Cambridge. Today I will give a talk on our new paper, BAND, Biomedical Alert News Dataset. This paper is collaborated by researchers from the University of Cambridge, University of Glasgow, and McGill University. We've developed a dataset to enhance the distribution of biomedical news available on GitHub. This resource supports advancements in public health informatics. Today, I will detail BAND's development, structure, and its significance in health communication. Let's dive in. In the natural language processing, we are faced with a significant challenge, the scarcity of sophisticated epidemiological datasets. Our solution to this problem is the creation of the Biomedical Alert News Dataset, or BAND. BAND comprises 1,508 carefully curated samples, each accompanied by 13 epidemiology-related questions. This rich composition allows for advanced research and application in health informatics, aiming to fill the current gap in data-driven epidemiological studies. In the ongoing efforts to keep an eye on infectious diseases, we rely on systems like Biocaster, GFIN, ProMed-Mail, HealthMap, and ELS. These systems are great at detecting potential outbreaks. However, they tend to focus more on the initial detection rather than detailed analysis. Currently, we still cannot catch everything, especially in complex situations or with groups who need more attention. We are also looking to improve how we use data to train our machine learning tools, so we can get better at finding and understanding disease patterns. The BAND dataset is a collection of 1,500 pieces, including news, emails, and alerts. Each piece comes with 30 questions that dig into health-related events. Our goal is to make it easier for computers to understand and use this information to help track diseases. We focus on the important bits that matter to people who study diseases, helping them to act fast when outbreaks happen. In our benchmarking process on the BAND dataset, we focus on 3 key NLP tasks: question answering, named entity recognition, and event extraction. The goal here is to rigorously evaluate and assess the capabilities of state-of-the-art NLP models. By doing so, we can determine how well these models can understand and process complex epidemiological data, which is crucial for advancing the tools used by health professionals in disease surveillance and response efforts. For this section, we'll dive into the process of data annotation. This is where the raw data is transformed into a structured format that's invaluable for our NLP tasks. This is the data annotation process. We start by carefully selecting the questions that will guide the annotation, ensuring they are relevant and comprehensive. Then, we proceed to choose the samples that will be included in the dataset. The annotation itself is carried out carefully, with each data point being tagged and categorized for ease of analysis. This step is crucial as it directly influences the reliability of our dataset. After annotation, we conduct a consistency check to ensure uniformity across the dataset. Finally, we perform an ethics check, an essential step where we ensure that the data collection and annotation process align with ethical standards. This thorough process results in a dataset that is not only rich and varied, but also trustworthy and ethical, setting a new benchmark for data quality in the field of NLP for epidemiology surveillance. For the question selection in our data annotation process, we've engaged with leading experts in epidemiology and public health. We've categorized these questions into 3 types: event-specific questions that track the spread and impact of diseases, epidemiological questions that delve into characteristics and patterns of outbreaks, and ethics questions to ensure the responsible use of data. In the sample selection phase of our data annotation process, we collect raw news alerts from ProMed-Mail, accumulating a total of 36,000 alerts. To ensure the highest relevance and quality of data, we employed an expert scoring system where samples were evaluated and prioritized based on their significance to epidemiological research by domain experts. For the annotation phase of the data, we utilized the Label Studio interface, which provided a streamlined and efficient way to tag and categorize our data. We partnered with a professional annotation company to ensure that the work was carried out to the highest standards. After each annotation batch, we conducted thorough reviews and incorporated feedback, ensure that each piece of the data was annotated accurately. In our commitment to accuracy, the consistency check is a crucial step in our data annotation process. We employed 5 annotators to work on the same set of 14 samples to ensure that the data labeling was not subjective, but rather uniformly understood and applied by all. This was followed by a manual review process to guarantee that the annotations were consistent across different annotators. The quality check is a vital part of our data annotation process. It involves a thorough manual review of the annotations to identify and correct any errors, ensuring the data meets our high standards. We also incorporate feedback from domain experts to rectify any misunderstandings, creating an iterative feedback loop between annotators and experts. This process refines the outcomes and ensures our datasets' accuracy, reliability, and utility for NLP tasks in public health. The ethics check in our data annotation process is essential to ensure our practices align with the highest standards of research integrity. This began with a thorough research ethics review, including obtaining the necessary permission from the faculty's research ethics committee. During the annotation phase, our annotators rigorously assess each sample to confirm compliance with ethical guidance. Any data that doesn't meet this criteria is responsibly removed. The band dataset provides an extensive overview of disease and location distribution, crucial for understanding global health patterns. It includes a wide array of infectious diseases like anthrax and cholera, offering a comprehensive view of health threats. Location-wise, the dataset spans a broad spectrum, encompassing a diverse range of countries, provinces, and cities worldwide, which underscores the data's global applicability and relevance in tracking and analyzing disease spread. Furthermore, the band dataset provides an exhaustive pathogen distribution, covering bacteria, fungi, protozoa, and viruses, ensuring a broad understanding of infectious agents. While it mainly focuses on human and animal diseases, it also includes plant diseases, expanding its utility across different fields of biomedical research. The dataset's symptoms include a vast array of symptoms that are instrumental for training models to detect and interpret disease indicators accurately. To optimize the band dataset for various research purposes, we've implemented 2 distinct data split strategies, rand split and stratified split. The rand split involves random partitioning of the data, providing a general-purpose dataset for a variety of tasks. The stratified split, on the other hand, is designed to focus specifically on sparse questions, ensuring that even the less frequently occurring data points are adequately represented and evaluated in our analysis. In this section, we will dive into the experiments conducted using the band dataset. We've tailored specific NLP tasks to maximize the utility of the band dataset. For the NER task, our objective is to identify critical entities such as disease names, outbreak locations, pathogens, and symptoms. In the QA task, our aim is to accurately answer questions using both extractive and abstractive methods. Finally, for the event extraction task, our focus is to pinpoint and extract relevant information about disease outbreaks. This is the NER task results. We found that supervised models have superior performance compared to the zero-shot model like ChatGPT. The possible reason for ChatGPT's lower performance could include the novelty and the specialized nature of our dataset, which may not be well represented in the ChatGPT's training data. Additionally, ChatGPT may formalize entities which could affect its ability to recognize them in the specific context of our data. In QA tasks, decoder-only model like Bloom perform better than encoder-decoder ones. Bloom excels likely due to domain-specific training and controlled output. ChatGPT shows lower performance, possibly due to its zero-shot application on new datasets and limitations in varying inference tasks. This highlights the need for careful model selection and potential fine-tuning for specific datasets. A detailed review of QA task results show ChatGPT sometimes misses possible inferences from data and occasionally over-infers compared to human judgment. For example, ChatGPT might correctly identify a city but not infer the associated country. This analysis highlights the importance of ongoing model training and refinement, especially for tasks needing deep inference and contextual understanding. In the event extraction task, models vary in performance by category with lower scores in province code and city code. BART excels in geocode predictions but has limitations in other areas. ChatGPT, in a zero-job setting, outperforms other decoder-only models, indicating its strengths in generating contextual relevant information without specific fine-tuning. Today's presentation covered the Biomedical Alert News Dataset, BAN, which consists of 1,500 samples and 30 event epidemiology-related questions, offering a valuable resource for NLP in public health. We examine tasks like NER, QA, and event extraction using models ranging from CRF-based to advanced ones like Loom and ChatGPT. Our findings highlight the challenges in NLP for epidemiological data and the importance of selecting appropriate models for specific tasks. The BAND dataset sets a new standard for future research, advancing AI's potential in public health. Thank you all for your attention and engagement throughout this presentation. We invite you to explore the BAND dataset further and contribute to its growth and improvement. The dataset is available on GitHub and the provided link, and we welcome collaboration from both the research community and the public health domain. Thank you.